Chapter 1, History. Some historically important events and people in the history of microbiology. In ancient times, many people had the concept that there were invisible entities or things that caused disease. What they lacked was the technology to confirm that these such invisible creatures existed. We have the advantage today of having technologies that can be used to confirm that there are invisible microorganisms that cause disease. Around 430 BCE, an ancient physician by the name of Hippocrates noticed that certain diseases were transferred from person to person, and that there were various routes through which diseases seemed to be transferred, whether it was sexually transmitted, respiratory infections. Of course, he didn't call them infections at the time, but certainly identified them as diseases or lack of normal function for those certain parts of the body. Hippocrates was quite famous. He's now considered to be the father of medicine and established many concepts and wrote many books, even 2,400 years ago when he practiced medicine. About 100 BCE, or about 2,100 years ago, a Roman physician by the name of Marcus Trentius Varro also noticed that people could transfer disease from one person to another and proposed that there were tiny, invisible animals that were the cause of these types of transmissible diseases. Again, the right idea, but no technology to confirm if their ideas were correct or not. By the time we get to the late 1500s, new devices called microscopes were invented. Move forward a few more decades, and we get to the mid-1600s, and a very famous scientist by the name of Robert Hooke designed and developed a microscope used to enlarge really small structures into larger images was perfected. It really is the foundation of the kind of microscope that we use today. There are similar parts and pieces. We have an ocular or an eyepiece that we would look through at the top, an objective magnifying lens at the bottom, pointed at the specimen that we want to look at, and even a focusing knob to alter the distance of the objective lens from the specimen to get us a nice, sharp, crisp image. Robert Hooke, when he was looking at different kinds of specimens, noticed that looking at a thin piece of cork, the little individual components and compartments in a thin piece of cork reminded him of the little cells that priests and monks would use for meditation and prayer. So he coined the term cell to mean those individual little compartments. We still use that term today to mean the exact same structures. What Robert Hooke saw when he used his microscope, in addition to identifying small structures that we now call cells, he also looked at all types of different insects, pieces of leaves, various other types of materials, including molds. Now, technically, Robert Hooke was the first person to look at the fine structures of a microorganism. The microorganism was a mold. If we take a look at some of the pictures that he drew in his book Micrographia, which he published in the 1660s, we can see very fine detail of things that we call molds, insects, and even parts of leaves using his microscope. Anton van Leeuwenhoek was a cloth merchant who worked with a simple microscope at roughly the same point in time that Robert Hooke was using the Hooke microscope. Now, the Hooke microscope is a compound microscope, uses two or more magnifying lenses. That's how we define a compound microscope. The microscope that Leeuwenhoek used was a simple microscope It has one magnifying lens, and if you take a look at the photo of the simple Leeuwenhoek microscope, compare it to the photo or the drawing of the Hooke microscope, you can clearly see there's a huge difference between the two. Now, Anton van Leeuwenhoek really is the first person to see bacteria with a microscope. Even though Robert Hooke technically is the first person to see a microorganism in the form of a mold, Leeuwenhoek did something different with a microscope that no one had ever done, and that's to simply use a microscope to look at other things. He took rainwater from a rain barrel, samples of material between somebody's teeth, little parts and pieces from someone's body, just all kinds of things that no one had ever viewed. He drew pictures of what he saw because no one had ever seen anything like that before. And sure enough, he described most of these tiny little things that he saw, because he could see that they moved, animalcules. Leeuwenhoek was quite astounded to note that there were so many little animalcules that he could view from the scrapings in someone's mouth that they seemed to be more numerous than all the people in the kingdom, and wrote down such an observation. He clearly did see the different kinds of shapes and sizes and structures that we now clearly today call bacteria. Another historically important person for us is Carolus Linnaeus, or Carol von Linnaea is sometimes another way his name is written in different textbooks. He is the individual who proposed a two-name system for the classification and identification of all organisms. Originally, Linnaeus was working with plants. He was a physician, and as a physician, he had to also be a botanist. Why do you suppose, in the 1700s, which is when Linnaeus worked, you had to also have a degree in botany to be a physician? Well, the two-name system is the binomial nomenclature system that we still use today. Binomial, B-I, means two. Nomial is referring to names. So this is a two-name system, and often the terms are usually derived from Latin or Greek, and we still use that system today. In the 1830s, two scientists, Theodor Schwann, who was a zoologist studying animals, Matthias Schleiden, who was a botanist studying plants, independently took a look at what were the fundamental structures or building blocks that made up plants and animals. What they determined was that the fundamental structures that were found in organisms that we call plants and animals were cells. So in 1839, they both proposed this, what we now call the cell theory, that all living organisms were made up of cells. We still use this theory today, and it's one of the 5 or 6 theories in biology that we'll discuss during this section on the history of microbiology. A concept that was very popular and many people believed true up till about the mid-1800s was a concept called spontaneous generation. Spontaneous generation basically says that organisms or living things could grow from non-living materials. Today we would call this a theory of abiogenesis, if in fact it were true. It was thought that if you took a piece of mud sitting on a pond or the bank of a lake, after a while that piece of mud would turn into a frog. Or if you had a piece of meat sitting out in your kitchen, that spontaneously that dead piece of meat would turn into living flies. There was even a famous experiment from the 1700s by a fairly good scientist by the name of Van Helmont. Van Helmont's experiment of spontaneous generation simply took a jar, some old cloth or sweaty underwear at the bottom of the jar, and then you lay a nice layer of wheat on top of that. Put that jar in a corner in your home, and after about 3 weeks, mice would emerge spontaneously from that jar. Of course, they were sort of missing some bits and pieces of information. What they, of course, lacked was the knowledge that mice are nocturnal, so they do all of their running around at night, and that open jar with the food and the nice dirty cloth was an ideal nesting site for mice to go and have little baby mice. Well, regardless, spontaneous generation was a concept that was difficult to disprove, and even was an underlying concept of how infection or disease occurred in people. It was thought that if you were sick, that the illness was a spontaneous one, and either due to the fact that you were a bad person, you did something wrong to displease God or some of the higher powers. There are all types of explanations as to where disease organisms or diseases came from, and also spoilage organisms. If you made a nice pot of stew or soup and you let it sit out in your kitchen, even covered, eventually it seemed to transform into a spoiled pot of soup or stew. Well, where did the spoilage organisms come from? It was thought that they spontaneously occurred or grew from that cooked food. We now know that that's not true, that that's not where the microorganisms come from. But how do you actually disprove that?
One of my favorite websites. The Metamorphosis of Frogs according to Aristotle from Piles of Mud. Seem to explain spontaneous generation.
In the 16 and 1700s, Italian scientists were trying to disprove spontaneous generation. Francesco Redi was one such scientist in the 1600s who tried to set up a procedure or experiment to demonstrate that spontaneous generation did not occur. It was well known that if you hung fresh raw meat in your kitchen or at the butcher shop, that eventually maggots and flies would appear on the meat. Well, lots of folks actually thought that the maggots in particular spontaneously grew from that dead piece of meat. In 1668, Francesco Redi set up an experiment using several jars and some raw meat. In some of the jars, he put the raw meat with no covering, and in other jars, he put a very thin piece of cloth. Now, leaving them sit for a bit of time, he was able to demonstrate that only the open jars had flies and maggots show up. The jars with the very thin covering of cloth, no maggots, no flies. That tried to explain or demonstrate where did the maggots and flies come from. Did they grow out of the rotten meat spontaneously, or, as we now know, that the flies come in, lay tiny microscopic eggs that start to develop into larvae that we call maggots, and that's where the maggots and the flies come from. Well, the critics at the time stated that one of the reasons that there was no spontaneous generation occurring in the jars that had cloth on the tops was that there was some vital living force that could not get through the cloth, and that because these were sealed containers, even though they were just loosely sealed, this vital force which was required for spontaneous generation could not pass through, and that's why no maggots or flies showed up. Well, it took another 200 years for scientists to establish a procedure that would thoroughly disprove spontaneous generation and answer all of the critical comments or ideas surrounding spontaneous generation.